Recombinant DNA technology can be used to combine DNA from different sources for DNA cloning or expressing proteins not found in that cell. The DNA of interest is obtained from genomic DNA, reverse transcribing mRNA, or a DNA synthesizer. Complementary DNA or cDNA from reverse transcribed mRNA is used instead of genomic DNA when expressing a eukaryotic gene in a prokaryotic cell. Prokaryotes do not have the molecular machinery to splice out introns, the non-coding region found within a gene in the genome, as prokaryotic DNA, like cDNA, does not have introns. Thus, using genomic DNA in bacterial cells for protein production may result in a sequence of amino acids that is not the protein from the gene of interest. Nucleic acids are extracted from cells by breaking open the cell and destroying or separating all undesired molecules. When you extract the DNA or RNA from cells, the genes or specific RNA will be extracted with all other genes or RNA found in the cell. So it may first be necessary to create a DNA or cDNA library, which is a collection of host cells with different fragments of DNA inserted, then isolate the host cell with the fragment of interest from this collection of host cells. Libraries, once created, can remain as permanent reference sources. For the fragment of interest to be copied and maintained, the fragment must be joined to a vector with an origin of replication. Examples of vectors are plasmids, bacteriophage DNA, or yeast artificial chromosomes. The choice of a vector may depend on the insert size, the inserting purpose, and the preferred host cell. Small inserts may be required for Sanger sequencing, so a plasmid vector would be suitable. However, large inserts with linked markers for identification cannot be carried by plasmids, which can hold a maximum insert size of about 20 kilobase pairs. Thus, different vectors, such as artificial chromosomes, which can carry large inserts, are used. If the gene inserts are to form proteins, an expression vector is used, which contains elements such as a promoter and a ribosomal binding site. If expressing eukaryotic genes in bacteria, the expression vector provides bacterial regulatory signals. Vectors need to be cut so that DNA can be inserted, while chromosomal DNA needs to be cut, as often it is too big to be transported by the vector. DNA is cut with endonucleases such as restriction enzymes selected based on recognition sites near the fragment of interest, or the restriction enzymes are not selected for use if it has a cutting site within the gene or cuts the vector more than once. Restriction enzymes can make staggered cuts in the two strands of DNA such that the cut ends have a 2 to 4 nucleotide single-stranded DNA called sticky ends. Restriction enzymes can also cut across the complementary DNA strands at the same position and create blunt ends. If the same or different restriction endonucleases which produce complementary or near complementary ends are used to cut the vector and source DNA for the fragment of interest, the vector and fragment of interest anneal through complementary base pairing. If restriction enzymes which form blunt ends cuts the DNA and vector, though there is no complementarity between ends, vector and fragment of interest can be joined, but the chance is lower. If the fragment of interest is to be inserted in a particular orientation within the vector, different restriction enzymes may be used to cut the vector as well as each end of the fragment of interest. The direction of the nucleotide sequence to the promoter is important when orientation determines if the protein is expressed, and if expressed, whether the sequence of amino acids is of the protein wanted. Cutting the vector with two restriction enzymes also prevents the cell circularization of the vector. The vector DNA and DNA fragment, even once annealed or connected, have gaps between the adjoining ends, which must be sealed with DNA ligase. Ligation occurs between the 5' phosphate and 3' hydroxyl ends of the different DNA to form recombinant DNA. Recombinant DNA lacks the machinery necessary for replication and gene expression and thus needs to be inserted into hosts such as bacteria, yeast, or mammalian cells. The host cell of choice is often bacteria, as these cells are easily maintained and manipulated. But when necessary, for example, when post-translational modification of a eukaryotic protein is required, a yeast or mammalian cell will be preferable to the bacteria that cannot modify eukaryotic protein. The introduction of the recombinant DNA into a host cell may be by transformation, transduction, or transfection. Transformation is the uptake of genetic material from the environment by bacterial cells. Transduction occurs when foreign DNA or RNA is introduced into bacterial or eukaryotic cells via a virus or viral vector. Transfection is the process of introducing foreign molecules into a eukaryotic cell through chemical and physical methods. The recombinant techniques are not 100% efficient, so some cells will not have vectors, others will have self-circularized vectors, and some will have recombinant vectors. Host cells with vectors are identified by some detectable property the vector's gene provides, such as antibiotic resistance or fluorescence. The host cell used must not have the gene for this property to differentiate the cells with and without the vector. If the selectable marker is an antibiotic-resistant gene, growing the cells on media with that antibiotic will identify cells with the vector. However, it may not be the recombinant vector. To isolate the cell with the recombinant vector, one would insert the DNA fragment into the vector such that it destroys the integrity of one of the genes present. Then, recombinants can be identified because the characteristic coded by the inactivated gene is no longer displayed by the host cells. One example is the inactivation of a second antibiotic-resistant gene. In this case, replica plating may be necessary. Replica plating involves pressing a velveteen-covered disc and then imprinting secondary plates, so producing identical patterns of bacterial colonies on a series of Petri plates. It is necessary to have the replica plates since the cells with the recombinant vector have the gene for antibiotic resistance fragmented and are identified from their inability to grow in the media with the second antibiotic. To recover the cells with the recombinant vector, they can be picked from the same location on the master plate. Insertional inactivation of the ligase gene may also be used to select host cells with recombinant vectors. An insertion within the ligase gene prevents the host cell from synthesizing beta-galactosidase. As beta-galactosidase metabolizes X-gal in the cell growth media to yield a blue compound, it is the white bacterial colonies that have the ligase gene inactivated. That is, it is the white colonies that have the recombinant vector. Since in media with X-gal there is no obligatory growth advantage, that is, you are not looking at cells that can survive but are looking for particular phenotypes, the process is called screening, not selection. The genes used for selection or screening vary in different host cells. In yeast, genes such as HIS3, LEU2, TRP1, and URA3 are used to locate host cells with vectors and recombinant vectors. So to summarize, recombinant DNA technology to clone DNA or express genes involve first, obtaining DNA fragments of interest from extracted genomic DNA, reverse transcribed RNA, or synthesized DNA. Second, using an appropriate vector such as plasmid, bacteriophage DNA, or artificial chromosomes for different insert sizes, host cells, and purposes, whether cloning or expressing genes. Third, using suitable restriction endonucleases to cut vector and DNA of interest, based on the availability and location of recognition sequences and the requirement for a specific insert orientation. Fourth, joining vector and the fragment of DNA with DNA ligase. Fifth, introducing recombinant DNA into a suitable host, be it a bacteria, virus, yeast, or mammalian cell, through transformation, transduction, or transfection. Sixth, selecting or screening for the cells which has the recombinant vector introduced. Now let's check our understanding of recombinant DNA technology with a few questions. You may pause the video while you think about your answer. If your answer tallies with the correct answer provided, well done. If not, you might have to view and listen to the video again. Question 1. You discover a novel eukaryote organism that glows in the dark. You believe this trait is due to a single gene and you wish to clone the gene. Which of the following strategies is most likely to be successful? A. Isolate the genomic DNA from the organism. Digest with restriction endonuclease. Insert into a plasmid vector and transform into bacteria. Screen colonies for the ability to glow in the dark. B. Isolate the genomic DNA from the organism. Digest with restriction endonucleases. Insert into a plasmid vector and transform into eukaryotic cells such as yeast. Screen colonies for the ability to glow in the dark. C. Isolate mRNA from the organism. Reverse transcribe and generate cDNA. Insert into a plasmid vector and transform into bacteria. Screen colonies for the ability to glow in the dark. D. Isolate mRNA from the organism. Reverse transcribe and generate cDNA. Insert into a plasmid vector and transform into eukaryotic cells such as yeast. Screen colonies for the ability to glow in the dark. The answer is D. As it is a novel organism, there is a lack of information such as restriction cutting sites, so it is better to use mRNA. Also, since the source organism is a eukaryote, the protein may require post-translational modification, which prokaryotes cannot carry out. The protein required for expression of the phenotype may only be produced by a eukaryotic host. Question 2. Diagrammed is a section of the human genome showing a gene of interest, regulatory sequences, and locations of recognition sites for 3 common restriction enzymes, EcoR1, BAMH1, and NCO1. In the vector shown, you would like to express this gene in E. coli for further study. What restriction enzyme would you use to clone this gene? A. EcoR1, B. BAMH1, C. NCO1, D. Either EcoR1, BAMH1, or NCO1. The answer is C. While the plasmid has restriction sites for all 3 restriction enzymes, EcoR1 cannot be used as it cuts the plasmid twice, so we would wind up chopping out a piece of the plasmid, including the needed Shine-Dalgarno region. BAMH1 is also wrong because it cuts in the middle of the gene we want to clone. We want an enzyme that will leave our gene intact. NCO1 is the best choice. It will cut our gene out of the genome and makes a single cut for inserting it into the plasmid. It's not important that the enhancer and TATA sequences will be left behind, as the plasmid provides the needed regulatory signals. Question 3. You plan to clone the gene of interest into the vector shown. The restriction sites available on this vector are also shown. Which restriction enzymes would you use to create a recombinant vector that would have the highest chance of expressing the gene of interest in the host cell? Why? The answer is the enzymes 2,1 and SAL1, as cutting with these restriction enzymes allows the insert to be positioned in the correct orientation to the promoter. Question 4. Which features in a vector facilitate gene cloning? A. Origin of replication only. B. Origin of replication and cloning site. C. Cloning site and selectable marker. D. Origin of replication, selectable marker, and cloning site. The answer is D, as the origin of replication is required to replicate and maintain the plasmid in the daughter cells. A selectable marker is used to isolate the host cell, which has the recombinant plasmid, and a cloning site is necessary to open the vector and insert the fragment of DNA into the vector. Question 5. Viral vectors typically can carry much larger pieces of DNA than plasmid vectors, but plasmid vectors are much easier to work with since they do not kill their host cells. In which of the following situations would you anticipate preferring a viral vector? A. You need to clone an entire gene, including the regulatory sequences. B. You are generating a genomic library from an organism with a large genome. C. You are creating an mRNA library to identify the gene encoding a very large protein. D. All of these. E. None of these. The answer is D, as indicated by words such as large in each of the descriptions and the description in the question which mentions that viral vectors can carry larger fragments than plasmids. Question 6. If you use a vector with the ligase gene as a screenable marker, which bacterial strains would you want to use as host cells? A. A bacteria carrying the wild-type ligase allele. B. A bacteria lacking the wild-type ligase allele. C. Both of these bacteria would work. D. Neither of these bacteria would work. The answer is B. As if the host had the wild-type ligase allele, you would not be able to differentiate a host cell with vector from a host cell with a recombined vector. Question 7. I am interested in cloning a piece of human DNA in the plasmid shown here using the SPH1 restriction endonuclease. To detect the DNA, I make 3 replica plates, minimal media plus tetracycline, and minimal media plus ampicillin. The description of the colonies I should keep are: A. Survives with minimal media and tetracycline, dies with ampicillin. B. Survives with minimal media, ampicillin, and tetracycline. C. Survives with minimal media and ampicillin, dies with tetracycline. D. Never survives. Note, minimal media contains the minimum nutrients possible for colony growth. The answer is C, as the recombinant plasmid will not have an intact tetracycline-resistant gene to express tetracycline resistance, but the ampicillin-resistant gene will be intact and able to provide ampicillin resistance to the host cell. Thank you for listening. More videos on genetic topics are available on this channel. Do subscribe to be notified of new videos.